All right, so this, like I said, this chapter is, the focus is on DNA and how, what I'm going to look at is specifically how it's copied, okay? Why? That's the topic. So, why? Because at some point, some people got together at the ministry and said, what should the grade 12s learn? And they made a list of things, and DNA replication is one of them. So we're going to talk about it. All right, so we actually talked a little bit about, very little about DNA in the section on biochemistry. So we know a little bit about it. Now this chapter here talks about, there's a lot of the history of discovery of DNA structure. So I'm going to get you guys to look at that. And I'm not going to spend a lot of time on that. So there's going to be stuff in this PowerPoint that I'm going to kind of skip over. I'm going to get you guys to read it over. We will go over it if you have trouble with it, but I'm not going to spend too much time on it, okay? So, trivia question. In 2003, there was something that was officially published. There was a rough draft that came out in 2001, but the official publication was 2003. It was a publication of, guess what? Very important publication. There's a hint on the black. There's a hint on the screen. The human genome. Jeez. Now, how many of you know what that means? Yeah, you would have been born. You would have been how old?
I would have been 5.
You were 5, so at 5, you guys don't study the genetics, stuff like that? What did you guys study in grade when you were 5? What was that, JK, SK? Grade 1? What is that?
JK is like square peg, round hole.
Don't pick your nose with a dirty finger? Yeah, okay.
Yeah, you're right.
Yeah, alright. So, so it's over a decade ago, the human genome was published. Now, what they were doing is they were racing, there were a couple of companies that were competing to basically decipher the entire human genome, which is basically all of the letters in your DNA. So if you look at a cell and you zoom in on the nucleus, you know that there's your DNA. And what these companies were doing is they were trying to decode that genome, the strings of A's, T's, C's, and G's. And the human genome is quite large. It's about 3 billion base pairs of letters. So you know the code of DNA, right? It's A's, T's, C's, and G's. We already looked at this. So hopefully this is not new to you. So what they discovered is that the human genome is quite large. It contains about 20 to 24, maybe 23,000 genes. If I wrote 21,000 genes, that's just an estimate. But it's really interesting. If you look at your textbook was published before the Human Genome Project was officially published. And your book estimates that we have about 45,000 genes. If you look at an older book might have said something like 100,000 genes. So what you see in biology textbooks is that over time, the actual number of genes has been reduced. It was estimated to be really big. Now we know it's closer to about 20,000. Why do you think the estimate was so big? Why do you think the estimate was so, we're off, the earliest textbook said about 100,000. The actual documents that were published show that closer to 20,000. Why? That's off by quite a bit. Why do you think the number was so big? How many of you know how many genes a fruit fly has? No? About 14,000. So why do you think our estimate was 100,000? Because scientists are looking at us fruit flies thinking that we're way more complicated than fruit flies. So we must have way more genes. Turns out we don't have a lot more. We have maybe 6,000 or 7,000 more, but it's not like 84,000 more or 86,000 more. It's a lot less than they thought. So what's interesting in the human genome is that they're still uncovering these genes because they basically have the list of A's, C's, T's, and G's, but now they're trying to go through it and trying to figure out, or find genes that maybe they haven't found yet. So, they're uncovering these genes. Maybe you guys, when you go to university, you might want to get into some genetic research. No, not even. Don't even want to do that, eh? So, does anyone know what the first gene discovered was? So this gene was actually discovered before the Human Genome Project was published. It was a gene for Huntington's disease. You ever heard of that gene? Yeah, you heard of that disease, Huntington's disease? You guys studying grade 11 a little bit?
No.
They also discovered, before the Human Genome Project was published, they discovered the gene for cystic fibrosis. You guys studied that in grade 11?
Getting a sheet on the count.
Was that?
Getting like a paper and to fill it out. No, then no.
Why is it important to understand this? Oh, wait, hold on. Let's pause the recording here. Because you got to understand, when they were sequencing the human genome, it took years. It took many, many years. And it cost, I would imagine, hundreds of millions, maybe billions of dollars to do it. Why would there be such an effort from a standpoint of people investment and money investment to sequence the human genome? Why was there such a push to do this project? It was a big thing to go to space in the 60s, and it was just as big a thing to go into and discover and decipher the human genome. Why? What's the big deal about knowing the strings of A, C, C's, and G's in your body? Who cares, right? Really? You wouldn't want to know about the genes that are working in your body? Why would you? What would be the advantage of understanding the genes? Okay, preventing what? All right, maybe studying a gene and trying to figure out why it causes disease, see if you can maybe figure out a way to prevent it, or maybe even cure it in the future. Maybe if you can figure out genes that promote regeneration, maybe we can figure out how to regrow body parts. You know that a few years ago, scientists figured out a trick. They figured out a way to take your regular skin cells and turn them into stem cells. Did you know that? That's kind of cool. But so if you can understand what these genes are doing, you could look at disease, you could look at prediction of disease, you could look at prevention of disease, you could look at maybe fixing genes, you could look at maybe repair of the human body, right? So it's very important understanding our genetics. And we'll talk more about this as we go through this unit, okay? So what else is cool is that not only do we have about 20,000 genes, but we have almost as many broken genes or dead genes, and they're called pseudogenes. So a pseudogene is a gene that no longer works. It used to work in the past, and we'll talk more about these types of genes later on in this unit. But staggering amount that we have almost as many broken or dead genes as we do working genes. Okay. We are 99% genetically identical. So there's a lot of things that came out of the Human Genome Project. One is that we are almost identical. There's only a 0.1% difference between Charlot and I, okay, on average. Now, but that 0.1% actually translates into quite a bit, because 0.1% means that if I take Charlot's DNA and I take my DNA and I look at a list of 1,000 letters, 1 in 1,000 would be different. Only 1 in 1,000 would be different. Okay, but how many letters do we have? 3 billion pairs, right? So that means that 3 million pairs would be different, because 3 million represents 0.1%. So even though we are almost identical, there is still quite a bit of difference between you and I, right? That's good for you guys. You don't want to be like me, right? So the human genome, only 2% of it actually codes for something. Now, do you guys understand how the basic idea behind genetics? We actually talked about this. The DNA encodes what special molecule? It's the molecules that do everything in your body. Starts with a P. The proteins. Okay, the basic idea behind genetics, and this is a very basic idea, is that DNA encodes genes. Sorry, in your DNA there are genes, and the genes encode these things called proteins. And the proteins do things in your body, right? Like they make hemoglobin or collagen, for instance. And only 2% of your DNA gives you protein. So that means the other 98% is doing something totally different. And we'll talk about that as we go through this unit. Okay. Now, the human genome, I said, remember the original prediction was that we have all of these genes. It turns out to be not true. We actually have less genes than a soya plant on average. Okay, so a plant that doesn't go to school, doesn't worry about what they look like, okay, doesn't have any attitude like you guys, has more genes than you. That's quite fascinating. Okay, we are over 98% identical to chimps. So our closest living relatives are chimpanzees. So again, if you take a stretch of DNA and you line it up with different organisms, chimpanzees are the ones that are most similar to us. Now this number is probably not 500. It's probably closer to 300 or 300 and something. If you looked at all living things on this planet, whether it's human, bacteria, chickens, or Jerusha, there are about a few hundred genes that all living things have in common. Okay, and we say that these genes are universal because we can find them in all of these living things. Okay. Now I don't know about the term. To me, when I say universal, it means like across the universe, but obviously on Earth. So whether it's bacteria or a human, they have these certain genes in common. Why would that be the case? Why do all living things share these several hundred genes? They're basically the same genes, whether it's bacteria or humans or foxes or soya. They have these sets of genes that are common throughout all living organisms. Why is that the case? Nika remembers her grade 11 biology, because we all stem from a common ancestor, right? And because we're all connected, because all living things are connected to each other, then we expect to see some similarities like this. Okay. All right. Now, I said in this presentation there's a whole bunch of stuff on the history of DNA. So this presentation talks about the history, but also talks about how DNA is copied, okay? The history part, I want you guys to look at it yourself. And I will go over it with you, okay? So like there's some experiments in here, like Griffiths transformation experiment, where I'll mention this one, but the rest of them you guys can look at. So you got to understand that we take it for granted that you guys know, and you probably taught this in elementary school, that your inheritance comes from DNA, right? When you look at a characteristic like eye color or hair color, we know that the molecule that determines that characteristic is your DNA. But if you go back 70, 80, 90, 100 years ago, they weren't really sure. They weren't really sure what the molecule was that was responsible for heredity. Okay? They thought maybe it was protein. The two leading candidates were protein or DNA. Right? So the history of, discovering DNA as the molecule for transmission, I'll let you guys look at that. I will mention this one experiment because it is kind of a cruel but neat one. So this is called Griffiths transformation experiment. And what this scientist did, Frederick Griffith, is he took two types of bacteria. So two types of pneumococcus bacteria. And one is called the R version, and the other one's called the S version. It's just by the appearance of the bacteria. R is rough, so it looks like it has rough appearance. And the S one is smooth. And when mice were injected, one killed, the other one didn't. Which was a lethal bacteria? Yes. Yes, the S. The S one was lethal. So bacteria have this trick. They can actually pick up DNA from other bacteria. They can undergo a process called transformation. Now we can't do this. Okay. For us to do this, for us to pick up DNA from some other organism, we need like a virus to inject it into our body. Okay. And actually, our genome actually shows that we have DNA that is actually being brought in from viruses. So here we have an experiment that shows this principle of transformation. So what happened was Frederick Griffith took that information where we know that R cells are safe and S versions are lethal, and what he did was he killed them. So now remember, the S one is lethal, right? So what happens if you take the lethal bacteria and you heat it, and then you inject it? What happens to the mouse? The mouse lives. Okay. So that makes sense because you know that if you take like water, if you're out camping and you drink water from a lake, before you drink the water from the lake, what should you do? What should you do with the water? You should boil it. Why? Right, kill bacteria. So nothing special here, right? But what's interesting is, let's think about this for a second, okay? He killed S as what? Safe or lethal? Safe. What was the R version? R version was safe, right? So the R version is safe. The he killed S is safe. What's interesting is when you mix the two together, what happens? Well, skies. So this told Griffith that the R cells, which are not being killed by heat, are being transformed. Now he didn't know what the molecule was. He didn't know that it was DNA. All he knew was that there's something that's being picked up by the R cells that is changing their programming. Okay? It's allowing them to become lethal. It's transforming them in some way. Okay? So this experiment shows that there's a molecule that can transform. Well, they didn't know what it was. So in this PowerPoint, there's a whole bunch of other experiments that talk about looking for this molecule of heredity. So again, I'll leave it up to you to look at that, and we will go over it if you need to go over it. So, like, there's the Hershey and Chase experiment, where they looked at viruses. I will leave it to you to look at, okay? I want to show you this chart. This is an important chart. So here's a breakdown. This was done by Erwin Sharga. Okay? He looked at the composition of because you know that there are 4 bases in DNA, right? Adenine, thymine, cytosine, and guanine. He looked at the composition of the 4 bases, and he noticed something. What do you see? A is very close to percent composition to what letter? T. And C is close to what? Yep. So look at here. Look at A. Whatever A is, T is very close. Okay? Whatever G is, C is very close. And we'll talk later about why it's not exactly 100, like exactly the same. And now you know the reason for this, right? You know that the reason for this is because of complementary base pairing. You know that like for example, C pairs up with. So here are the bases, right? Here's cytosine, thymine, adenine, guanine. You know the reason for that is because A pairs up with what? T. So if A is 20% and A pairs up with T, then T should be what? 20%. So as long as you know one of the bases, you kind of already know what the other ones are. Like if A is 20%, what's G? What must G be? If you look at the genome of an organism and you know that 20% of it is adenine, then what would G be? What would guanine be? Well, if A is 20%, thymine would be what? 20%. That's 40%. What does that leave you with? 60%. So that means 30% must be what? G, and the other 30% must be cytosine, right? Because of a complementary base pairing. Now, do you guys know who actually put together the structure of DNA as a molecule? Yep, it was Watson and Crick. And so they're the ones who actually put together the structure of DNA. And we know that it's a double helix molecule. We know that it comprises of 4 bases, A, C, T, and G. And we know that A pairs up with T and C pairs up with G, right? Okay. Now, this is important because this actually, when Watson and Crick put together the model of DNA, it actually told them a lot. Not only what it looks like, but actually told them how it gets copied. And the model is so simple. It's really, really simple that when it was published, scientists said, oh, how stupid of me not to think of this myself, because it's such an easy idea to understand. So let's talk about how, and by the way, here's a list of the, no, I said, like, you're going to look at the history, right, on your own. Here's a list of experiments and discoveries that led to the conclusion that DNA is the molecule that is responsible for heredity. So again, you can look at that, and we will talk about that if we need to. Okay. So let's talk about DNA replication. So you're going to learn about 3 big ideas in this unit. Okay. The 3 big ideas in this unit are replication, transcription, and translation. Replication is DNA to DNA. Okay, so when we make copies of DNA. Replication, transcription is DNA to RNA, and translation is RNA to protein. So today we're going to introduce replication. Okay, so the idea of replication is really, really simple. The the idea is you take a strand of DNA and you make 2 copies of it, and then from those 2 copies you can make 4 copies. And then we'll talk about mutations. We'll talk about what happens if you make a mistake. And we'll spend a whole chapter on, not a whole chapter, but we'll spend a whole day on mutations, okay? So let's talk about how DNA is copied. It's a really simple process. What I'm going to do is I'm going to start with an animation, okay? That's going to show you the basic idea of how DNA is copied. Okay? It's really, really simple. So, here is a strand of DNA, okay? And you can see that complementary base pairing, right? You can see that that C pairs up with G and A pairs up with T, right? Now, there are a bunch of enzymes that are going to be involved. Okay, you'll learn their names and what they do. But for now, I want you to just focus on what's actually happening, okay? So, take a look at this animation. I'm not going to explain anything. All right. Now, in your own words, I haven't explained anything yet, okay? What happened? Yeah, I know, but what happened though? No, don't give me any names of enzymes. Just in your own, what happened? Take a look. What does this remind you of? Yes. So it's splitting, it's unzipping, right? And what do you think the big idea is? The big idea is you take a strand of DNA, you unzip it, you open it up. Okay? And then what you do is if you open up DNA, you have a single strand over here, and you have a single strand over here, right? And then what do you think you do with that single strand? Do you copy it? You pair it up with what? Its complement. So if you take DNA and you split it, right? If you take a DNA molecule and you open it up, you have single strands that are exposed, right? You have a single strand over here, and you have a single strand over here. And then if you simply insert the complement, so if you have an A, you put in a T. If you have a G, you put in a C. If you see a T here, what would you put in here? An A. If you do the same thing on this side, if you have a G, you put in a C. If you have an A, you put in a T. What do you end up with if you do this? Take a look. What do you end up with? I want you to look at these 2 strands. What do you notice? Are they the same or are they different? They're identical. So DNA replication is really simple. Okay? You take a strand of DNA and you unzip it, you open it up. You use each strand to build the complement. And if you do that, you end up with 2 molecules that are what? Exactly the same. Okay? That's the big idea. Now, as we go through it, you're going to see it's a little more complicated than that because there's a few things that are happening. Like, for example, you'll notice that we actually don't even start with DNA. How many of you noticed that? Did you catch something? If you look at this animation, you'll notice that you're using a red color here for some reason, followed by a blue color. And you'll notice that if you look at the red color, you'll see a letter in there that doesn't belong. It's the letter U. Now, U is not found in DNA. Where is it found? RNA. So DNA replication is simple, but there's a few things that we have to talk about later on, okay? So one is like, okay, why is there RNA? Why do we need RNA in this process? Okay? There's another thing I want you guys to consider. Look at the way it's being copied. You'll notice that this strand over here, which way is it being copied? Right to left, this way, or left to right? It's going which way? So let's back it up. So take a look. Take a look at the top strand. Which way is it going? It's going this way, right? But look at the bottom strand. You notice it's going the, why is that? That's weird. Why is one strand going one way? Why is the other strand going the other way? That's very odd. So there's a few things that we'll talk about. I mean, DNA replication is very simple. Don't get me wrong; it's simple, but there's a few things that we need to worry about. So let's just talk a little bit more about replication, and then, by the way, what I'm showing you, that's an animation. Why do we use animations in biology? What's that? Because, well, it's to show you like an idea, right? So that it makes it easier to understand it, right? But animations sometimes are too simple, right? Because we have to simplify it. Otherwise, if you make something really complicated, you're not learning anything. It's going to be like very confusing. So I will show you later on actually what DNA replication looks like. It doesn't look like that. It looks a little bit different than that. Okay? But not today. Okay. So here is your strand of DNA. Now, you'll notice something about the way they're oriented. If I said to you that DNA is an antiparallel molecule, you know what parallel means, right? What do you think antiparallel means? What can you give me an analogy? Can you give me an analogy of something that's antiparallel that you experience every day in your life? So parallel means they're moving the same way, not perpendicular, right? But antiparallel means that they're moving the opposite way, but they're parallel. So can you give me an idea of something that maybe you use every day that's antiparallel? What? The roads, right? When you're on the road, right? You have one road that's going one way, and beside you have the other roads going the other way, right? The roads are parallel, because if they weren't, that would be a big problem, right? And when we do get the perpendicular roads, that's an intersection, right? Okay, so you'll notice that this strand is going this way, and then this strand is going this way, okay? And, but the idea is, and we'll talk about the importance of this, not today, okay? So, in replication, I said the idea is very simple. You take a strand of DNA and you put in the what? Complement. So if you're reading a strand of DNA, you put in here a T, this would be a what? A G, right? Here are the molecules that are involved in DNA replication. We will go through these. Okay, we will go through these. This is a neat little picture of DNA actually opening up, because as you saw in the animation, the DNA has to unzip, right? And the reason why it has to unzip is because if you think of DNA like a book, you have to open up the book to read it. So we need to unzip it so we could expose the single strands so we can copy them. And that's what you're seeing here, okay? So this is called a replication bubble. This over here and this over here are called replication forks. What's a fork in the road? When you can go two different ways, right? So you can see a fork here, then you can see a fork here. You can actually see in this animation that there's another bubble over here in this corner. I can't see the whole bubble, but I can see the fork. Okay? So this is the molecule that actually opens up the DNA, because there has to be an enzyme that actually unzips it. This is it right here. This is the molecule helicase. This is a protein. You can actually see that it changes between two different shapes. And as it changes between these two different shapes, it actually unzips the DNA molecule. So to me, it kind of reminds me of like a pair of scissors that open and close. So helicase is the molecule that actually unzips. So here is actually a strand of bacterial DNA. Now, their DNA is different than ours. Theirs is actually a big loop. It's a circle, okay? So here's a strand of DNA, bacterial. You can see that it's being unzipped. So it's opening up over here. And that bubble just gets bigger. Eventually continues to grow. And then you end up getting two molecules that are exactly what? The same. Okay? Because remember, the idea is the single strand is used to build the complement, and then you end up with two that are exactly the same. Our DNA is a little bit different because it's not a bubble. Sorry, it's not a circle. Eukaryotes, so in prokaryotes, the DNA is a circle, but in eukaryotes, pretty much anything that's not bacteria, the DNA is a line. You can stretch it out, and you actually have more than one bubble. So here are actually two bubbles. Eventually, two bubbles will merge together, but the idea is still the same. The idea is you take the DNA, you unzip it, and you use complementary base pairing to build a new DNA molecule. Okay. So here's a picture of this. Here are some of the enzymes that we'll talk about next time. So here, remember that red section? That's the same thing over here. Okay. Here's helicase. This is the enzyme that unzips. Okay. Now, this is where I'm going to leave off. Okay. So the basic idea behind replication is what? You take DNA, you unzip it, you open it up, right? And then you take the strand and you add the complement. And if you do that, you read the single strand and you build the complement, you end up with two molecules that are exactly what? The same. So this section and this section should be exactly what? The same. The molecule that's going to do the copying, the reading and copying, is called DNA polymerase. Now, I think I showed you a picture of DNA polymerase before. It's an enzyme that makes DNA. Okay? Yeah, I think I even asked you a question about because, this is a protein, right? DNA polymerase is a protein. And where does the information come from to make that protein? Remember, all proteins are encoded in what? DNA. So here we have a protein that is encoded in the DNA, but makes DNA. Remember that I asked you the question, which came first? Remember that question? Do you know where the answer was? No? Okay, we'll talk about it later then. This protein is, and this is actually the active site of the protein. This is a beautiful picture, one of my favorite. You can actually see in this picture the secondary level of a protein. You can see the coiling. You can make up the coiling here. This is the active site. The strand that is being read in DNA is called the template strand. So right now, I can see that this is the nucleotide that's being read. This is the one that is being inserted. And you see the letter C here? So what we're inserting is the letter C for cytosine. So therefore, what must this template, what must it be? This has to be a G, okay? So this is a neat little picture looking in the active site of DNA polymerase. This I can see is magnesium. So what do we call a molecule that an enzyme uses to help it work? Remember what that we call it? A cofactor or a coenzyme. Now in this case, is it a cofactor or a coenzyme? What do you think it is? This is a good review for you guys, for your test. What do you call an inorganic molecule, like magnesium or iron, that helps an enzyme function? Is that a cofactor or coenzyme? It's cofactor, yeah, very good. Whereas vitamin C is an example of a coenzyme. So I thought I would end with this because a neat little picture of inside the active site of polymerase, and then on Thursday we'll continue. Okay? All right, ladies? Okay, let's end there.